So, hi everyone, my name is Jingyou Yu, currently a postdoc from Dr. Dan Barouch Lab from the Beth Israel Deaconess Medical Center and the Harvard Medical School. So our lab right now has our lab has long-term interest in studying the immunology and the virology of HIV infection, as you know, and developing novel vaccines and some of the eradication strategies against HIV. And now we are also applied our vaccine expertise to other infectious diseases of those global significance, such as the Zika virus, the tuberculosis, and most recently now the COVID-19. And for me, I'm now mainly focused on the SARS-CoV-2 vaccine development and helping with our team to develop better vaccine strategies right now.
Could you sort of describe the timeline that you worked with to develop this new vaccine for COVID-19?
Yeah, so it's kind of interesting because at the very first time we were first encountered this COVID-19 vaccine, so we were in the lab retreat back in the 2020. I think the January 2020. So we were in the Museum of Massachusetts. We were having like kind of the lab retreat. So Dr. Dan Barouch kind of discussed with us about this newly a series of the around 40 infected people in China, in Wuhan, and we're discussing possibly maybe we can develop a certain kind of vaccine to target the SARS-CoV-2. At that time, we do not know the exact sequence, so we're trying to wait till the sequence is released, then we can work on that. So that's the first time we formed this kind of idea to develop this vaccine, and I'm very fortunate. Actually, at that night, I remember it is a Friday night, so we look into the website and see that one of the Chinese group has released the whole SARS-CoV-2 sequence on January, and we quickly just get started on to develop this novel vaccine, design the sequences and put into the vectors and send to the company to synthesize the genome. The vaccines, and so. And then quickly we just quickly we want to develop certain kind of an animal model. There is a kind of a we don't want to experiment this vaccine in the humans directly. We want to use animal model to determine the safety and immunogenicity as well as the protective efficacy. So we quickly just first design the vaccine, and then on February we just immunized the models, include the mice model, the ferrets, the hamster, and most important, the rhesus macaques. So that's in February. We immunized these models, and we determine how could this model useful to measure the SARS-CoV-2 infection, as well as how good it is to determine the vaccine the efficacy. So on on May, we got our final data, and we just summarized the data and publish our papers in Science. So first paper is describing the pathogenesis caused by SARS-CoV-2 in the monkeys, and the second one introduced the DNA vaccine we developed to see how good the vaccine is in the monkey models, and quickly. Once we have that good, results, we quickly collaborated with Johnson & Johnson, and we initiated actually the phase 1 and phase 2 clinical trials on July, and on September, so we also initiated a single shot the phase 3 clinical trials, which just enrolled more than 40,000 people participants. And on November as well, we initiated another 2 shots phase 3 clinical trial, which enrolled more than 30,000 people. So eventually on this year, so in January, so FDA applied, just authorized the emergency use of the F-26 based vaccine, and now actually a lot of people are just taking the shots from the different clinics. So I think it is a very unique experience for us.
Could you describe some of the underlying science that has made it possible to develop this in such a short time?
For example, for us, because for other vaccines, I'm not quite sure, but at least for the adenovirus vectors, there are several, there are years of research on the adenovirus vector vaccines. So originally we have like Ad5, which is a prototype advector. So based on that, there are several clinical trials has been initiated and conducted. So we have gathered a lot of information about that. And later we have a novel where even improve the platform like Ad26 or other like from the NHP adenovirus vectors like what the AstraZeneca used, like the chimpanzee advectors. So those has been has been greatly improved, and based on that, we just step by step, based on that knowledge, so we are developing better and better vaccines about that.
Is there anything else that is cool about your research that you want to share? Or
as I mentioned, the most exciting thing is once you spend the weeks, month, and even 1 or 2 years efforts, you finally see your efforts turn out to be like a product, and that product is useful and is functional. Because sometimes you can publish something, or it always can be highlighted in a newspaper, but in reality, when you see someone or benefit from you or from some of your efforts, that's make you feel kind of very special and very, that's for, at least for me, it's kind of very exciting moment for me. If you see the picture, someone are taking your Johnson Ad26 vaccines, you see, they kind of like, once they take the shots and they feel like more confident, you see, okay, you definitely, you do something significant, you do something useful. So I think that from that part, I think that's most important and my most cool thing for me.